Hey everybody, we've got another review kind of mind map thing here as we get ready for the exam. This is going to be molecular genetics. I'm going to call this part one. And because molecular genetics, there's just a ton. And so in this one, we're going to look specifically at structure, and we're going to look at replication and transcription, just again, at a very, very high level. This is not meant to be an instructional video. This is really review. So to start off, we got to look at our structure of DNA. Remember, DNA is deoxyribonucleic acid. So our backbone has a deoxyribose and a phosphodiester bond into a phosphate. If we kind of sketch this, let me do this over here. So there's a phosphate group that's attached to a deoxyribose and a nitrogenous base in the middle, attached to the next phosphate. This bond right here is a diester bond, so we have single O like that. So an O, single bond O is an ester, so if we have two of them, you have a diester bond. So when we are looking at the deoxyribose, we're talking about DNA. RNA is just ribonucleic acid. Same structure, it's just a ribose. It's got all of its oxygens on it. In this inside of your DNA, remember it's a helix. It's a double helix if we're talking DNA. A pairs with T, G pairs with C. C and G bonds are triple hydrogen bonds, and remember it's just an interaction, an attraction between them. So these guys take more energy than A and T do, just because A and T only have a two hydrogen bond interaction inside that ladder. So when we're breaking apart the DNA, either in replication or something else, G and C take just a little bit more energy. This is a highly regulated process. There are a lot of regulation mechanisms here that mainly keep the DNA organized. We're always fighting against mutations, so we do not want mutation, right? That is like enemy number one when we're talking genetic code. So there's a few mechanisms that we use. First is methylation. Methylation keeps the DNA attached to the histone. So when we wind this DNA up, it wraps around a histone complex, and those histone proteins just provide some structure to our chromosome. Remember, our chromosomes, each one, our code is about 4 billion bases long, so there's a lot of organization that goes on there. And on that 4 billion bases, we have about 10,000 genes, and we have to know where they are and what they do, and so they are wrapped around these histones and methylated to keep them structured correctly. Acetylation, something similar, but it helps unwind the DNA. So when we acetylate it, we loosen that DNA off of the histone just a little bit. We can get in there with enzymes and other proteins that we need to carry out some kind of task, whether it's to duplicate, replicate the DNA, or whether it's to transcribe a gene into a protein. We've also got transcription controls when we're looking at eukaryotes versus prokaryotes. So eukaryotes, we have these things called promoters and enhancers. So I've got a simple diagram over here. So here is a simple eukaryotic gene. On one region upstream from the gene, we have a promoter. Then we've got our gene. And then somewhere downstream, we've got these transcription factors, or enhancers. And these enhancers, these transcription factors, can be anywhere on the code. They could be on the same chromosome. They could be thousands of base pairs away, again, because we are trying to avoid mutations. Like, this is the entire thing. So by isolating different regions of the DNA, we can control against changes to the DNA. So in eukaryotes, we have transcription factors and enhancers that actually fold this linear strand of DNA back when these transcription factors attach to the promoter. So you got to imagine here, we've got these transcription factors attached to this thing. Then, and only then, can our polymerase attach and begin to transcribe that gene. That this gene becomes active. If these transcription factors fall off, then the gene becomes inactive, and the polymerase will fall off as well. So it's a control factor here to make sure that we only produce this gene when we need that gene to be expressed. Prokaryotes are just a little bit different. Prokaryotes use this thing called an operon, and there's some structural difference. So first of all, in an operon, the control factors are all linked right next to each other. So there could be an enhancer region that would have some kind of activating agent, like a ligand. There's the operator, and then there's the promoter region. The operator is what actually turns the promoter on or off to transcribe a gene. These three things are always in sequence and always upstream from the gene when we're looking at prokaryotes. I have two videos, or one whole section on operators and promoters, so I do encourage you to go back and look at those. But the main thing about these is that they are feedback-based. Oops. So if we have a positive feedback system, we need the presence of something to activate that operator, to remove some kind of inhibition. If it's negative feedback, the presence of that molecule will actually inhibit that gene from being expressed. So go back and take a look at those videos. I'm not going to dive into the whole thing here. We're also going to look at there's 4 major enzymes that are involved in replication and transcription. So helicase, when we're talking about helicase, we're talking about replication. Helicase is this guy in the diagram. So it follows the DNA, or it runs along the DNA, and it actually unzips it, and it causes what is called a replication fork. So you can see we've got 2 strands of DNA here. Pay attention, these are anti-parallel. So the 5' strand is up here, and the 3' strand is down here. Helicase just facilitates the unwinding of that DNA so that our next enzyme, our primases, and actually I have these out of order, so if we're looking at replication, this is going to be number 1 here, and then we're going to sit number 2, 3, and 4. So helicase unwinds the DNA strands, then primase comes in, and it simply lays down a primer. And that primer is a little piece of RNA that goes on the DNA strand, and it is laid down from 5' to 3'. So my anti-parallel strand, this is a 5' piece of RNA, and here's a 3' piece because I have to be opposite my template strand. Same thing up here. This RNA primer is 5' to 3' because our strands are anti-parallel. The next enzyme to come in is DNA polymerase. Remember, we name things in science, obviously. So polymer, I'm building a polymer, and it's an ACE, so that means it's an enzyme. So this is an enzyme that polymerizes or builds DNA. The DNA builds 5' to 3'. So on this bottom strand, this is the leading strand because it can follow right behind the helicase. It is building the 5' end of the DNA and just chugging along as it builds. The top strand, this is called the lagging strand because it has to leapfrog backwards. So polymerase will build 5' to 3', and then leapfrog to the next section that opens up, and these are called Okazaki fragments. So that's transcription. That happens on DNA when it's replicating itself. So if you think about your cell cycle diagram, so let's draw our circle. We've got mitosis is the tiny little slice, and then we've got the 3 other large chunks. So this is G1, S. This is where replication is occurring, right, in this chunk here, and there's some other control factors there. And then ligase comes in last, and ligase cleans up. It removes the primers. It binds the DNA strands together. It also proofreads a little bit as we're going. So these 4 enzymes, they do a lot of different things, but mainly we're trying to make a perfect copy of our DNA. Transcription is a little bit different. In transcription, we have similar enzymes, except there's no helicase in transcription because the RNA polymerase, so we have RNA polymerase, so we're building RNA. RNA polymerase is like a giant self-contained factory. When I'm building a protein, I only want to transcribe a gene, right? So this would be like a portion of a gene. That gene is all I care about, so I'm not going to unwind my entire DNA strand to get to that gene. So RNA polymerase comes in, it actually unwinds the DNA itself, and it reads a template, 3 to 5, and builds 5 to 3. So this is the start of the mRNA that is being produced by the polymerase. And as it reads along, it fills in the complementary base pair. So if we zoom in, you can see we have T here, T pairs with A. The next one is an A. In RNA, there is no thymine. We have uracil, so this guy pairs up with adenine in the DNA. C pairs with G, G pairs with C, and so on. As that mRNA is produced, it exits the RNA polymerase. So the RNA polymerase, first of all, it's ignoring this top strand. It's reading this bottom strand, reads from 3 to 5. So it's moving in that direction, and it spits out mRNA in that direction. And that's what we're going to talk about in this video. So we've talked about the structure, we looked at replication and transcription. The next piece, when RNA is produced, it moves out of the nucleus and is translated by the ribosomes, and we'll look at that in the next video.